Hey, good day, everyone. Thanks for tuning in. Today, I'm going to be talking to you about some new research I've been working on, a new neural network architecture called HyperXPair, which is designed to learn distance-dependent motif interactions. The outline for this talk is as follows. We'll start by describing what the biological motivation for this work is. We'll then talk a bit about how CNNs can be used to discover so-called biological motifs. I'll then present what I call the distance-dependent motif interaction problem and state what that is clearly. I'll then present to you the HyperXPair architecture, which we propose as a solution to the distance dependence problem. I'll then review some of the experiments that we performed and the results that we got. So this work here is motivated by an interest in how DNA ultimately becomes proteins, or what is often referred to in biology as the central dogma. This is a very complex process that involves a lot of moving parts, many of which are these so-called RNA binding proteins, or proteins that are capable of binding to DNA and RNA at specific locations, usually called something like a motif. And they coordinate together in a very specific way in order to do work inside of the cell. There are two case studies I'd like to discuss before going into the details of how the method works. The first is transcription initiation, which is a process in which DNA is turned into an RNA transcript. This generally involves the cooperation between two motifs. One of them is the start codon, which is the part of the RNA that becomes protein, and that has the motif AUG. But there's another motif that's upstream or to the left of that start codon called the Shine-Delgarno sequence, which is often given something like GGAGG. It's the combination or concert of these two motifs that initiate transcription, and having one without the other won't result in a valid protein. Now, what's interesting about the cooperation between these motifs is that they're separated by a precise number of bases, but a variable number of bases. So anywhere from 3 to 15 bases apart. Beyond that, the Shine-Delgarno sequence isn't a Shine-Delgarno sequence. Shorter than that, it also is unable to serve its role. The second case study here is pre-mRNA splicing, or once that transcript is produced, it gets cut up and glued together to result in what's called a mature transcript to become a protein. This process is also mediated by a number of different RNA binding proteins, which bind to the RNA via motifs. One example of a motif involved in splicing is the branch point, which has itself a specific consensus sequence or motif sequence involving usually some strong A or UGA. And downstream to that, there's another sequence or motif that's important as well, which is an AG. And it's the combination or the co-occurrence of these two motifs that allow splicing to occur. Like transcription initiation, these motifs occur a specific but variable number of bases apart. Somewhere, I think, on the order of like 13 to 25 bases. Beyond that, splicing may not occur. So what do these case studies tell us? Well, they reveal two aspects of RNA binding proteins and motifs that make prediction quite difficult. The first, which we didn't touch on so much, is motif degeneracy, which is that some genomic events make use of fuzzy motifs. So while GGAGG is the prototypical Shine-Delgarno sequence, there are a number of variants of that sequence which can lead to the same result. So it's not so clear-cut. The second is that motifs interact. And this is clearly highlighted by both the transcription initiation and branch point examples, which is that some genomic events require more than one motif in order for them to occur. And not only that, not only do both motifs need to be present, but they need to be present in a certain context, i.e. separated by a certain number of positions, or else the genomic event may not occur. So the research question we have here is, can we train a model on known outcomes to learn the motifs and their interactions when the motifs and the interactions are unknown? For this, we'll use deep neural networks. The idea, in a very broad sense, is we have some DNA sequences, and we're going to put them into a model to predict some sort of outcome. In our case, we'll say the outcomes are already known. We already know if the transcript gets initiated, or we already know if and where splicing occurs. We don't know is why or how, so we're going to try to build a model to understand that. The idea of learning motifs is not new and has been done for quite some time and quite effectively using CNNs. The way this basically works is you take your DNA sequence and you one-hot encode it. So if you look at the top here, we turn the sequence, which has L positions, into an L/4 matrix. And in the first position, we have a C, so we put a 1 in the C column. The second position is a G, we put a 1 in the G column, and so on. We then pass a convolutional filter on top of that, which the weights being learned through the neural network, through backpropagation, learns in a certain way to represent some type of motif. And these motifs can then be combined deeper into the network to make the final prediction. When you have a single layer, you can very easily learn motifs, and you can predict the outcome as a combination of those motifs. It's interpretable, but it may not be adequate for all problems. Because of the complex nature of the genome and the fact that motifs often interact in very elaborate ways that we don't always know, it's more common now to build deep convolutional neural networks where the deeper layers of convolution can begin to capture these complex interactions. They're very powerful prediction tools. However, deep convolutional neural networks are black boxes. When you use deep neural networks, what happens is that the early layer motifs, those filters which represent the RNA motifs or the DNA motifs, they become fragmented and the motifs are no longer coherent. And the interactions, if they are captured, are completely opaque. They're buried inside of the neural network and we can't know what those interactions are. So while deep CNNs learn interactions implicitly, what I want is I want a model that can learn those interactions explicitly, including the context dependence or distance dependence aspect. So why exactly is it so difficult to learn motif interactions? Well, we know that an interaction means it's something more than the sum of its parts. But motif interactions depend more on more than just the presence of multiple motifs. But there's this element of context that somehow the distance between those motifs is also important. So the classical statistical interaction model, where you have an additive combination of the motifs plus some term which represents the product of the motifs, is not adequate because it doesn't capture the context element. We can try to define this problem a bit more carefully, and I'll call this the distance-dependent motif interaction problem. So given a sequence where motifs and the distances between them, which we'll represent as distributions, are unknown, we want to learn the identity of the motifs and the distance between them as a distribution. So we'll assume that if the motifs are within an appropriate distance, represented by a distribution, then the event occurs, which is y = 1. But if the motifs are present but too far apart or too close together, i.e. they're outside of that distribution, the event won't occur. We'll have a y = 0. The event also won't occur if one of the two motifs or both of the motifs are missing. This figure shows a schematic of a HyperXPair architecture. The input is its DNA sequence or RNA sequence, which we one-hot encode like we showed in the figure before. We then pass that one-hot encoding through an initial convolutional neural network, which can learn the motifs through filter weights. Those filters are those convolutional filters are applied over the one-hot encoding to get what I call a motif activity map. Or in other words, for each of the motifs, which we'll say there are F motifs, we'll get some value which represents how closely that motif maps onto that part of the DNA. So it gives us sort of a squiggle line where a peak in that squiggle says that motif is present at that position. Using this motif activity map, we'll apply a second convolutional neural network that's designed very carefully to capture motif interactions. And we'll talk about that on the next slide. The goodness of this motif interaction or motif co-occurrence will then get another number, which is like another squiggle line across space for each of the pairs. So while we had F motifs, we're now looking at F squared pairs. And there'll be another dimension that represents the relative distance between those pairs, where when S equals 1, you're talking about that one pair right next to each other, up to however far of a relative distance you want to consider. This information is then passed through a max pooling layer to give us a final score for each of the pairs, which are then used to predict the outcome, which is transcription initiation is occurring or not, or splicing is occurring or not. So the novelty of this architecture is the motif interaction module. And what we do is we assume that this context dependence or this distance dependence is defined by a distribution, more specifically a Gaussian distribution, that has the hyperparameters mu and sigma. And that distribution is unique for each motif pair, where there is some optimal distance between that pair, which is mu, and some variation around that optimum, which is sigma. And these are hyperparameters of the neural network. Using these hyperparameters, we can define for each pair a receptive field that we can pass along the motif activity map to measure to score the goodness of the co-occurrence. And this receptive field looks something like this, where we have one member of the pair is anchored at position 1, and the other pair follows some distribution based on the hyperparameters. As a technical implementation thing, we then take this and we break it up into what we'll call a 2-hot encoded kernel, but that's not really important at a conceptual level. That's more of a technical implementation thing. Then what we'll do is we'll train this model using a 2-step training procedure. In an outer loop, we're trying to optimize the hyperparameters, the best mu's and sigmas, the best intermotif distance distributions that lead to good accuracy. And we'll use that, we'll optimize that using Bayesian optimization. And in the inner loop, we're going to try to say, given this set, given this context dependent, given this distance dependence, try to find the best motifs. So we're learning the motifs and we're learning the distributions, but we're doing that in this 2-step training procedure. So the experiments we use to test this model, there are 2 that we focus on that mirror back to the case studies we presented before. The first is a transcription initiation, and for this, we'll use simulated data, simulating a binary classification problem. Either the DNA leads to a valid gene or valid protein, or it does not. This is a binary classification problem, and the way that it's defined is that y equals 1 when the Shine-Delgarno sequence and the start sequence are both present and have the correct distance. And there are many examples where either one are present or both are present with the wrong distance, and the neural networks needs to be able to tease these apart. In the second problem, we'll look at splice prediction, which is to say, can we, for a given segment of sequence, predict whether or not there is an acceptor site present or a donor site present within that sequence? So our results, we benchmarked it against a few different neural networks, some natural language processing methods, LSTMs, other convolutional neural networks. And we find that when we look at the simulated data, it performs really well. In fact, HyperXPair can perform as well as a deep convolutional neural network. because the problem here is defined exclusively by the distance-dependent motif interactions, and it can pick those up very efficiently. It's a little bit worse than real data, and this isn't so surprising because there isn't really a reason to assume that the distance-dependent motif interaction problem defines all of splicing, although we do believe it to be part of it. Nevertheless, even when this assumption is built into the model architecture, it's able to perform quite well. As I mentioned before, our primary objective actually is inaccuracy, though. It's interpretability. It's to build a model that can tell us something about how the biology is happening. And what this model can do, what our HyperXPair architecture can do, is it can tell us two things. It can tell us the identity of the motifs that were discovered, but also through the hyperparameters, through the mu and the sigma, which are themselves interpretable. It tells us something about the distance distribution that defines that motif pair. And what we can see here for the model applied to the simulated data, for which neither the motifs nor the distance distribution are known to the model, but are known to us because we did the simulations. The model is able to very quickly and very efficiently learn both the start codon and the Shine-Delgarno sequence. But not only that, because a lot of models can do that. What our model can also do is it can learn the distance between them as a Gaussian distribution to say that motif 2, which is the Shine-Delgarno sequence, is usually separated by the start codon by about 10 bases, plus or minus quite a few. And again, because this is simulated data, we know what that distribution ought to look like. And indeed, this is approximately the correct distribution. Now, when we apply it to the splicing data, we get some interesting results. What we find looking at the donor site is that it seems to have discovered the correct consensus here. Indeed, there is this sort of GGT, which is sort of part of this, what they call canonical donor site, or it's the consensus you would always expect to see. But it's also done something potentially quite interesting, which is if you look at the distribution, it said, we've got the second motif that is about 4 bases ahead of the first motif with very little variance as well. And what you see when you look at these sequence logos is that there's this TGAG at the end of motif 2 and a TAAG at the start of motif 1, which given the distance of 4 and these overlapping 4 base pairs seems to suggest it's stitched together 2 smaller motifs into a larger coherent motif. And this is an interesting finding because it suggests that HyperXPair is quite, has a certain robustness to the assumption of distance-dependent interactions as being important, that when that assumption doesn't hold, it can still piece together just a single motif if that's the most useful one in making a prediction. The acceptor, we get something quite interesting as well. What we find is in the first motif, we get a canonical AGG site, which is known to be involved in defining a splice acceptor site. And we also see a number of CCs with a couple Gs scattered about, sort of occurring upstream of this canonical site. Now, I'm not sure if this represents a specific RNA binding motif. I did try to look and cross-reference a few things in the literature and couldn't find any evidence that this corresponds to a specific RNA binding factor. We do know that upstream of the first motif is a polypyrimidine trap, and these Cs could represent that. It could be more specific, we're not really sure, but the real point of this model, but our intention here in designing this model was to be able to use neural network and machine learning to generate hypotheses about the biology. And indeed, here we have in these results an implied hypothesis. The presence of these 2 motifs separated by this number of distance is involved somehow in splicing. And we can take the data that we get from this model, having no prior knowledge, and we can generate a conceptual model of how splicing occurs, where we've got this idea of a donor site, we've got this idea of an acceptor site, And there's some pairing that's occurring here, some interaction that's occurring here when these two motifs are about 13 to 21 bases apart. And this now, as a conceptual model, can be something that we can validate through experiments or through referencing the literature. Great. Thanks so much for listening. If you have questions, please feel to reach out. I'm happy to talk about this further. Great. Thank you.